Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll answer your questions from the question and answer window, and your questions there will only be visible to myself and our speaker. So thank you for joining me today. It is now my pleasure to introduce our speaker, Tanner Nevill from Berkeley Lights.
Well, thanks for the introduction and looking forward to going through this deck with everyone. I will be making some forward-looking statements, so I've got a disclaimer up here for that reason. We'll start out right off with the fact that a central regulatory requirement for all cell lines manufacturing biologic therapies is monoclonality assurance. The gold standard for confirming that production cell lines originate from a single cell is still conventional well plate imaging. And unfortunately, well plate imaging is prone to error, largely due to the presence of so-called ghost cells and debris that are difficult to differentiate from cells. Now, this problem can be made even worse because you're not dealing with just a single well. You have stacks of 96 well plates that all have to be meticulously verified and tracked in order to ensure that clones are not misidentified. Ideally, you shouldn't have to worry about these ghost cells. You should be able to focus on developing cell lines. So at Berkeley Lights, we really like to solve problems just like this one, and here's one way we do it. Single cell technologies are well poised to replace manual human verification, providing a new standard for automated clonality assurance. So the Opto Cld Workflow on the Berkeley Lights platform provides a complete visual record of each clone at every stage, from single cell cloning through multiple days of on-chip culture and their assessment. The Opto Cld workflow begins with the automated loading of transfected pools from well plates onto nanofluidic Optoselect chips in the Beacon system. And I'll show some videos coming up on what the cells look like once they're in the system, but first let's have a closer look at these chips so I can show you what makes them so powerful. Each chip contains thousands of nanopin chambers. Each one is less than 1 nanoliter in volume, which is over 10,000 times smaller than a well in a single well from a 96 well plate. And this very small volume enables efficient growth and detection of secreted antibody molecules in less than an hour. Our Optoselect technology manipulates cells with light, and here we see a single CHO cell being automatically identified by the software and moving into a nanopin chamber. This light-based manipulation can be performed in parallel across hundreds of cells. And what that means is that thousands of CHO cells can be cloned into a single chip. The system can handle up to 4 chips at a time. So in less than 3 hours, we can load and screen thousands of individual single cells. And importantly, if multiple cells get placed into the same pin, automated cell counting enables the software to automatically detect those pins. They can be selectively removed and reloaded with single cells. So here we see chambers with more than 1 cell being cleared out and replaced with single cells. All of this is happening without any form of user intervention. After cloning single cells, cells are cultured on chip by perfusing media through the channels. Fresh media comes through and diffuses into each nanopin chamber, while waste products diffuse out and are swept to waste. This video shows an Opto Cld workflow where CHO cells are expanding on chip for 5 days. And something you may notice right away is the large diversity of growth rates and cell phenotypes, sizes, and morphology. This is all from a single transfected pool. Clones are imaged on the system throughout the workflow, providing visual confirmation of clonality. And in order to select top clones, you're going to need a sensitive way to measure the production of antibody. So in the Opto Cld workflow, we use a fluorescent small molecule named Spotlight. Spotlight gets imported into the chip and binds to the secreted antibodies in the nanopins. And when unbound Spotlight reagent is flushed out of the chip, the pins with the largest amount of secreted antibodies have the brightest fluorescent signal. So this assay is quantitative over a wide range of secretion rates. These assays can be performed at multiple time points over multiple days. And so over time, you can see the increase in signal, which reflects the growth of the clones. And because there's automated cell count at various time points, we can normalize the signal to the number of cells in each pin and extract a per cell productivity. We've spent a lot of time developing software for ranking and selecting clones based on these growth rates, assay results, and any other phenotypic measurements such as size or shape. In the video you're watching, each row depicts the entire clonal history for every single nanopin on the chip, starting from an empty pin to the loading of a single cell, followed by multiple images of cell culture and assays over multiple days. Filters can be used to select and rank clones based on clonality, growth rates, and any of the phenotypic measurements made. The last step in the process is recovery. Clones are automatically recovered based on their selection parameters. Cells are removed from pins using the same light-based manipulation you saw earlier, and then media is flushed to the channels to deposit cells into well plates for subsequent scale-up. Now let's further consider the need for clonality assurance. Our pins and chips have been specifically designed so that single cells can be identified unambiguously by on-chip imaging. So as a result, the hours of manual image verification are replaced by automated cell counting in software that's powered by AI. With the technology available on our Beacon systems, we're able to find the best clones and recover them with unrivaled clonality assurance. We have tested clonal recovery by alternating recovery of each clone with flushing media through the chip and into a blank well, and then imaging the well plates to see if any cells turned up in the blank wells where there should be no cells. The Opto Cld workflow does provide two different unload methods that enable clone recovery, but for each method, only 2 wells out of nearly 1,300 blank wells were found to contain cells. This translates to greater than 99% clonality assurance with extremely high yields. And the CLD team at Amgen recently published a paper in Biotech Journal which supported these monoclonality results. In this paper, Amgen also reported that cloning efficiency was significantly higher than using other methods, 52% on Beacon versus 11% for FACS and 5.7% for limiting dilution. So not only are they seeing significant improvements in the number of clones they're able to screen, they're getting extremely high monoclonality assurance rates. So the Opto cell line development on the Beacon system is one of the ways we solve problems because it enables you to select cell lines with greater than 99% monoclonality in a much faster way than typical methods. The clones can be selectively enriched for unique phenotypes. Multiple assays can be used to measure productivity of different types of antibody molecules. So thank you very much for your time. I look forward to any questions you may have.
Okay, great. So before we ask Tanner questions, we have some questions for you. You should see them now on your screen. The first question, if you can go ahead and use your mouse to select your answer. Apart from titer, what is the most important parameter for selecting a top clone? And the next question is, how many clones do you scale up into shake flasks and/or amber mini bioreactors? And the last question is, which product quality attribute is most important when selecting a production cell line? So again, if you can use your mouse and go ahead and select your answers from this screen. You can also type in your questions at this time, and we'll begin the Q&A shortly. We're just giving you a few more seconds to go ahead and answer the questions. Okay, great. We see a couple of you have answered. It's giving you a little time for a few more people to go ahead and select their answers on the screen. Okay, great. So the first question we have is for Tyler. Tyler, just making sure that you can, or Tanner, sorry, just making sure that I can hear you.
Yep, can you
First
hear me
question.
okay?
Yep, great. Okay. And what has the FDA said about clonality assurance?
Sure. So I can't speak to any specific IND filings our customers have been involved with, but I can say that our monoclonality data package has been shared with the FDA and received a positive reception. Additionally, multiple customers have validated our data package themselves, including Amgen, Catalent, and GSK. Amgen published their work in the Biotechnology Journal last year. Catalent has their work published in a GIN piece, and you can probably find that on their website. And GSK co-presented a webinar earlier this year with us, which you can find on our website. And then lastly, antibodies from our platform are in the clinic. SARS, a new neutralizing antibody discovered on our platform by folks at Vanderbilt University, have now entered phase 3 trials with AstraZeneca. And a production cell line producing a COVID-19 protein vaccine candidate was selected at the University of Queensland, and that's in clinical trials in Australia.
Great. So do the dimensions of the nanopen permit multiple layers of cells, namely CHO cells, to grow on top of one another?
With CHO cells, typically not. The height, kind of from floor to ceiling of the nanopens, is about 40 microns. And a CHO cell is, on order of 10 to 15 microns in diameter. And we typically don't see them stacking on top of each other. The chip is pretty much flat. And so gravity, these cells still behave the laws of physics. So gravity keeps them on the floor of the nanopen.
Okay. And has the FDA okayed the ME? I think you slightly addressed that, but.
Yeah, I think so.
Okay. And when assaying the titer, there were a few bright lights like stars on the screen. What are these?
In some cases, they are antibodies aggregating. And a question we often get asked is, can we detect aggregation of proteins on the chip? And I think we're working on that still. So there's some potential there.
Okay. Would you say this is comparable technology to Clonality and F-Site?
F-Site? Trying to see if I can read the question.
Question number 5 there.
I'm not familiar with F-Site. Clonality. Yeah, maybe you can ask that question a little bit more, and I'll try and answer it better.
Okay. And do you have any experience with suspension versus adherent cell lines?
We do. So most of the folks using our platform are working with suspension cells, but we do have a few folks working with adherent cells. So we do actually provide, it's a different surface chemistry. So we have the ability to actually coat the inside of the nanopens, but not the main channel. And so that allows for coating of an electrocellular matrix to allow adherent cells to grow in the pens and not anywhere else. And I realize I think I know the answer to the previous question.
Okay.
I believe you're asking about a single cell printer. And so those systems are designed to do similar to what a fax system can do, which is place a single cell into a well. So obviously there's not much comparison in terms of being able to screen for both production and growth rates, as well as other sort of morphology metrics like our system can.
Okay. And can you describe the risks of cells moving from chamber to chamber, especially during export of a chamber?
Yeah, so that we took a very, very close look at that. And that's part of the monoclonality data package I talked about. And you can reach out to us. I saw someone else asking, how do we get access to that? So we provide that to our customers. And so if you reach out to us, we can share that information with you. We have sort of exhaustive video detailing of these cells culturing over time, and lots of evidence to show that there is no travel from pin to pin to pin to pin during this process.
And how have you assessed the well-to-well cross-contamination, and what is the incidence of well-to-well contamination?
It's less than 1%. So when we quantify the clonality of the system, it's greater than 99%. And when we compare it to things like limiting dilution, it's equivalent to 4 rounds of limiting dilution with a half a cell per well.
And can this system be used for cells other than CHO?
Absolutely. Yep. I couldn't list all the cells that we've worked on this, but a large number of mammalian cells, and we're actually now doing a lot of work with non-mammalian cells, yeast and bacteria as well. The system is agnostic to cell type and media type, so it's really almost any cell type can be used with the system.
Okay. And looks like there's a couple more questions. You kind of addressed this already, but where can we find the BLI data package that was submitted to the FDA?
Reach out to us. We do only provide that to customers.
Okay. And did you encounter any cells that are really sticky in nanowells that are not easy to open?
Yes, cells come in all shapes and sizes, and we have seen lots of cells that can be sticky on chip. We can address it a couple different ways. There are some additives we found that play really well and mitigate the clumping. I'm sure folks have experienced this in other workflows as well. And we've actually just launched a sort of an upgrade to our export process that enables getting little stickier cells out a little bit better.
Okay. Well, it looks like we have time for just one more question. If we didn't get to your question, we'll be passing all the questions to Tanner, and he can follow up with you directly. So the last question is anyone using this for bispecifics?
Yes, definitely. So customers are starting to run CLD workflows on the Beacon with bispecifics and other non-standard mAbs. GSK recently presented some of this work. And again, I think that's actually on our website as well. And I'm aware of a few others. I can't really share the details, but absolutely yes. And currently we're providing reagents that bind to the FC region, as well as the kappa light region, and potentially more to come.
Okay, great. Thanks, Tanner. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us for future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.